ziba zisanzwe. So this PCR ikoreshwa mu bintu byinshi cyane indwara nyinshi, uretse n'indwara n'ibindi byinshi bibaho muri forensic cyangwa se muri clinical trials. The technique of PCR of course ikora amplification ya DNA. So the DNA first of all will be extracted from the cell. There is a process of DNA extraction. If you go to lab, there are a procedure for extracting the DNA from the cell. How you extract the DNA from the cell, how you destroy a cell, then after destroying it, you kill some other component like a protein and the others, and those component of the cells, you kill them, then you remain with the nucleus, and then you extract it from the nucleus, and you end up by having a copy of DNA. That's for example. Or RNA, you may also extract the RNA. But because this PCR will only amplify the RNA, then this RNA has to be transformed to DNA. You have to make a copy of DNA, what is called cDNA, or RNA reverse transcription. So it's making DNA from RNA, so that once you have a cDNA in your lab, then you can amplify it with the PCR. So PCR, you have two PCR. There is a real-time PCR. This real-time PCR is there for counting, and there is also conventional PCR. So conventional PCR is only for detection. The conventional PCR, you learn the gel, and you have the band which showing this DNA are present or not. So while the real-time PCR is amplifying, and then it will count and to give you the number. And the number, if you go to lab, I'll show you how the number there and how it's give it to as like a CP values. So PCR DNA can be amplified from single strand, that I said, but DNA can be amplified in vitro by cloning or recombinant DNA. Amplification of DNA by P is faster and technically less difficult than Replica's cloning method using a recombinant DNA technique. You will give you a homework course about the combination. The polymerase chain reaction PCR can produce many copies of specific target segment of DNA. I think I told you about this. So the 3 steps of DNA are heating, cooling, and replication. So the heating one is the PCR have 3 steps, which will involve cooling, heating, and replication. So this will so the heating is what is called the DNA denaturation. So in the 3 steps of DNA, one is the denaturation, annealing, and extension. So denaturation is separating the double strand of DNA by heating. So you heat, and then the strand of DNA will be separated. The key PCR is an usually heatable, stable DNA polymerase called tag polymerase. So for the DNA to start the elongation, we need the DNA polymerase. You know, we started about the DNA replication when we have seen that the DNA polymerase is the key for the replication. So for this to start the elongation, we will need the DNA polymerase. But because we are using heat, we need a polymerase which is resistant to the heat. That is why we need a tag polymerase. It's called the tag polymerase. Tag polymerase is obtained from a bacteria which is resistant from heat. It may resist up to 1,000 Celsius degree. This bacteria is called Thermus aquaticus. It's the bacteria where the its polymerase is structured. So this is heat stable DNA polymerase. That's why it's called heat stable DNA polymerase. So DNA polymerase used PCR is originally obtained from heterophilic bacterium called Thermus aquaticus, hence designated as tag polymerase. So this is the PCR in brief. As you can see, we have our sequence here, our DNA, and then the first step to be the denaturation. You heat at 90% and 90-degree, 94-degree heating for 1 minute, and then we separate into 2 strands, and then we mix a single strand DNA. There is a mixture which has the prime and which that mix is called must mix. So it has everything. So that's that in that mix we add the primers, which is strand DNA with attach the primer, and then we heat again at 72 Celsius degrees for the tag polymerase to start the extension, the elongation. And then we end up by having the first cycle, you see here, the second cycle, the third cycle, up to the so many cycles. So do you understand? Do you get something, students?
Not well.
Not well. So I told you that this PCR has 3 steps. PCR ifite step 3. Nababwiye yuko DNA igira ari DNA dukoresha muri PCR. Dushobora kuyikura muri cell, aribyo bita extraction of DNA. Cell ifite DNA, then in the lab we mix with some reagent zizagenda zikica za component zindi za RNA za cells, and then zigasiga DNA yacu. Iyo DNA tukabara ni zingahe dufite, ariyo counting DNA. Twarangiza tukaza muri PCR ndakeka mwabyumvishije ko PCR ibero gukora copies nyinshi. Ntabwo ko PCR ifite ubwoko bubiri, hariyo real time PCR na conventional PCR. Real time PCR ni ukugira ngo ikore copy nyinshi inazibare, itubwire ngo harimo copies zingana gutya. Umuntu afite copies zingana gutya za DNA wenda ya coronavirus cyangwa ya HIV cyangwa ya hepatite cyangwa ibindi bintu utazi, cyangwa malaria n'ibindi. Hakaba n'indi bita conventional PCR, iyo ni ukugira ngo igaragaze yuko iyo DNA ifite y'icyo kintu. Ni ukuvuga ngo yo ni ukureba tureba tukaraninga tukareba bande, iyo bande ko iyo ntuza tuyifite. Ni ukuvuga ngo ni gute noneho iyi PCR ikorwa. Dushobora gufata tuvuge nko ku gihe cy'iki ntuza cy'i COVID, dushobora ku extractinga RNA from patient sample, from patient za sample bagufata mu mazuru, and then tukajyamo cyangwa mu nkanka mu ntuza zimwe bafata mu kanwa, tugakuramo RNA. Dukoresha tukayuse different method reagent, for example triazole. It's one of the reagent that we are using to extract the RNA. Twamara kubona RNA tukazibara, and then kuko PCR templifying RNA tugomba kuzihinduramo DNA, aribyo bita cDNA, a copy of DNA. Ibyo bita cyangwa se reverse transcription. Twamara kubona izo DNA cDNA, noneho tukazikoresha PCR. PCR ikora uburyo butatu. So a template ya DNA yije, urabona ibi double stranded, ariko kugira ngo tubashe gukora transcription igomba kubanza igafunguka. ubundi tuzi ko mu buzima busanzwe ifungurwa n'iyi enzyme warayize yitwa helicase, ariko hano in lab ho dukoresha heat, we heat kugera kuri degree 94 za strand zigatandukana. Iyo niyo phase ya mbere yitwa denaturation, heating. So once they are separated, and then we start noneho endi yitwa annealing. Annealing ni ugushyiraho ama primers. Usanzwe tuzi ko muri transcription dukoresha primers. So in lab tuza dufite primers of a specific disease, of a specific DNA that we want to amplify. For example, if you have to amplify malaria, we have primers for malaria. And then you have a DNA there. And then once we mix and we amplify, tukaza kubona ikintu ubwo nukuvuga ngo malaria is afite DNA muri iyo sample. Niba ari covid ni gutyo. Kwangu niba ari covid, tukaba dufite sample y'abantu babiri mu ishuri ryanyu, tuvuge harimo umuntu witwa Françoise, akaba n'undi witwa Emmanuel. Bagiye kwipimisha covid, bo ntago bazi ko bayifite. So tumaze gufata sample from their sample tuzishyize hariya, and then twe extractinzemo DNA, turabona DNA turafite ariko ntituzi ngo aya ma DNA afite covid or not. Icyo gihe tufata za samples z'abo abantu babiri batandukanye tukazijyana muri PCR. PCR icyo ikora first ni denaturation, ikafungura za DNA za strand breaking down into two strand, one and another strand. And then tuzi yuko kugira ngo habanze haba transcription ibeho, twako kuba dufite primers. Kuko our target is the COVID virus, ubwo icyo tuyibuze gukora turakiramo primers za COVID kugira ngo turebe koko niba izo samples zishobora ku making a copy of COVID. Niba koko za primers, kuko primers iragenda ikajya kuri specific strand. Niyo muri za DNA strand za DNA harimo ya COVID, primers zizagenda zijyeho. Niba muri kwa France arwaye COVID, primers zizagenda zijye kwa ntuzo, kwa France. Yes, you're getting me? Muranyumva? Ya COVID, then the specific primer will come and attach on that DNA. Niba mu bya Maria ubundi murwayi nta kintu kirimo, the specific primer will not attach. And then at the last, the third step, it will be the amplification. So then the transcription irakatangira. Kuri wa muntu ufite COVID, primer kuko zafashe, so the RNA polymerase will come and start the transcription. Kubera ko primers zayo. Uwizafite zifite ko primers zitabaye attached, RNA polymerase will not act. And then it make a lot of copies of that strand. Turangiza noneho umuntu tuvuze ngo muri iyi sample tubonyemo kopi za COVID 3,000,000 n'urugero, mu yindi nta nimwe twabonyemo kuko wa wundi ntayafite, primer zabo zaho zifata, ariko undi kuko ayifite primer zabonye aho zifata. Ibyo nagira ngo mbasobanure mbabwire ko this PCR has 3 steps. One is denaturation, annealing and extension. Of course, the last one will be detection and analysis. So now do you understand?
Yes, we understand.
Okay, good.
Yes.
So the PCR is a very powerful diagnostic and research tool. In microbiology, it can be used to pick out the amplified DNA sequence that is unique inside the organism and enable the identifying, for example, HIV viruses with nabibabwiyeho. So there are also blood transfer techniques. So the blood transfer techniques used in visualization of specific DNA, RNA, protein among the thousands of molecules. These are, there is 1,000 blood. As transfer uses determine the number of copies of genes present in a uh, given tissue and a mutation in the gene. So for the mutation of the gene, we use southern blot. There is also northern blot, transfer technique used to size and quantify specific RNA molecules. So actually, the PCR is northern blot. So southern blot is for the tissue and the mutation. Northern blot is for RNA, RNA PCR, and then Western blotting is for the proteins. So the technique used to size and quantify specific protein molecules is called Western blotting. Western blotting is currently used as one of the tests for the HIV in the case of the present viral protein and blood detected in our bodies. So now you see the northern blot, southern blot, and the western blotting. So this is southern blot, so it's a DNA. Northern blot is RNA. So these two are PCR, is in southern and the northern. And the western blotting is the protein. So, but all these are based on the gel. I don't remember if you remember the transfer extracted from 1000 DNA